Hello, class. So for this particular lecture, we're only focusing on RNA metabolism, and it's not a series. This is the only video. And so the topics that will be discussed is DNA-dependent synthesis of RNA and RNA processing. So as far as the transcription by RNA polymerase, it is very similar, well, pretty much essentially the same as the mechanism used by DNA polymerase. And so for RNA polymerase, as a reminder of what happened in the lecture about DNA metabolism, we have the magnesium 2 ion. One is helping promote that free hydroxide group to do a nucleophilic attack to the alpha phosphate group, and the other magnesium plus 2 ion helps dissociate that pyro group. And as far as with the actual creation of the RNA, it utilizes the template of DNA. And remember, just like how DNA is synthesized is 5 to 3, the same thing with RNA. It is synthesized from 5N to a 3N. So therefore, one of the strands for the DNA will be the template strand, and the other strand will be a non-template because it goes in the wrong direction. Because it needs to, when you make your RNA, it needs to be anti-parallel to the DNA sequence. And so when you have this representation of the RNA polymerase, this purple figure, you have DNA that's going inside and it's actually being unwound. And while it's being unwound, in front of the transcription, you have this positive supercoil, and then you have this generation of RNA where you have an entry where the nucleotides can go in to start building up the RNA strand. And then from there, as the DNA comes out, it actually gets wound back together again, and you have this negative supercoil. And the direction of transcription is from left to right. And so just going into more details for the transcription, initiation, and elongation by an E. coli RNA polymerase. So you have your RNA polymerase, you have this certain code that's a promoter for having the generation of RNA, and then you have this cofactor, this sigma 70. And so both of them makes this complex with the RNA polymerase, where it is a closed complex, and then transcription bubble forms, where now that complex is now open and it starts generating the transcription for making RNA. And so from there, as the promoter, this moves out, you start to have elongation of the RNA being formed, and then eventually that cofactor, the sigma 7D, moves out and gets replaced with a protein NUSA, and so more elongation is happening, and then once this protein gets dissociated, that's when the RNA the transcription of it is terminated, and the RNA polymerase is recycled to do this process all over again. And so as far as the termination of transcription, there's two different methods. you have your P-independent termination, and that's when the polymerase bypass the site, or the complex undergoes a conformational change that ends up producing this RNA transcript that has a hairpin. And from having this hairpin, it disrupts the RNA DNA hybrid, and the RNA thus gets dissociated from the complex. And so this is the P-independent termination. And then the dependent termination, you have this P-helicase that actually separates the messenger RNA from the DNA template. And so this helicase migrates along the messenger RNA in the 5 to 3 direction and ends up separating it from the polymerase. And so that is the P-dependent termination. And so as far as dealing with transcription and processing, when you have a eukaryotic cell, so you have this you have this cap that is on this RNA strand, and so from there you have the exons, which is coding region that contain instructions for making proteins, while you have introns, which are non-coding regions that are removed during the RNA splicing before the messenger RNA is translated. And so you see that removal of the introns. And then from there, you have the termination of this RNA transcription, and you have your free RNA polymerase that's released and able to undergo transcription and processing again. So as far as this cap, how is that created? So over here you see this is the actual structure of the cap that's on the messenger RNA. You see highlighted here, you have 7-methyl guanosine, and then that is attached to the 5' end of the nucleotide. And so you can see, just make sure you can count. You have 1, 2, 3, 4, 5. That's why it's the 5 position. Also notated on the second position, you have methyl groups are often found at the second position of the first and second nucleotides in vertebrate cells. And so how is this cap being formed? You have these different enzymes that are involved, where the synthesis of the cap is carried out by enzymes tethered to the CTD of the polymerase 2. And so you have these enzymes, the phosphohydrolase that removes the phosphate group. You have the guanylyl transferase that release a pyrophosphate. Then you have guanine 7-methyl transferase that transfer adenosine methyl group. And so that's where that methylated nucleotide is coming from. And that happens twice. And then you actually have your end cap of that of the RNA. And so from there, the next step is actually the splicing. So to have the splicing to occur, you have in the first step, the hydroxyl group of the internal, right here, the internal branch actually attacks a phosphodiester bond at the fifth position right here, which results in a site cleavage, and the free hydroxyl group that was created also does a attack to the phosphodiester bond at the 3 position, which ends up releasing the introns, and that's how you have that termination of the introns. And this is the same sort of splicing mechanism, just kind of to notate where you have that free hydroxyl group that does a nucleophilic attack to the phosphodiester bond and thus releasing the introns. And now we need to talk about the actual termination. And when you have this termination of the RNA strand, there is actually a tail that is capped at the end. So once again, you have your direction of your transcription. You have your RNA being made, where you have your cap and you have your exon. There is actually a sequence for during the termination where you have AAUAAA endonucleus that also reads this termination type of sequence. And when it does see it, about 10 to 30 nucleotides downstream of that sequence is where it does a cleavage. And then from there, you have polyadenylate polymerase that actually creates a tail for the RNA, where that polyadenylate factor is removed, and ATP to help promote this dissociation, and thus ends up with this sequence of nucleotides that will be part of the tail of the RNA. And that concludes the RNA metabolism.